Today we are going to be talking about non-Mendelian genetics. So Gregor Mandel was a scientist. He was also a priest, and he looked at peas, so like sweet peas or green peas, and he found that peas could be wrinkled or smooth, they could be green or yellow, and by breeding them, just like we've done with our dragon game, he was able to figure out that some traits are dominant and some traits are recessive. But it turns out that most genetics is actually non-Mendelian. Very few genes have what we would call pure dominance or pure recessive characteristics. And the first one that we're going to look at today is called incomplete dominance. And incomplete dominance, in order to identify that something is incompletely dominant, you're going to look and see if, first of all, if there are three phenotypes. And the next thing that you're going to look for is you're going to look and see if there's sort of a middle version. So in this case, there's sort of like a full, a medium, and a light versions. Or in other words, there's a middle version. Of that trait. So we saw this in the game. So when we were looking at Drake armor, we saw that if you had full as your alleles, then you would have 5 armor plates. But if you had full none, then you would only have 3 armor plates. And if you had none, then you would have no armor plates. So this is like full armor, medium armor, light armor, or no armor. And there's this middle version where 3 is the middle between 5 and 0. That's a really ugly K. Let's just do a quick. There we go. Now this also happens in real life, and it happens a lot with plants. So when we're looking at flowers, this is not all flowers, but it is some flowers. We have red is dominant to white. So if I have a red flower, then that means that it has the big R genotype. If I have a white flower, because that's recessive, I have a little r genotype. But in the middle, between red and white, we have pink. And if I have a pink flower, then that means that you have 1 big R and 1 little r as the genotype. Okay, the next thing that we're going to look at often has 3 phenotypes as well, but you can have more, and that is multiple alleles. So in multiple alleles, you're going to have 3 or more phenotypes. So you might be thinking, oh, I've got something with 3 phenotypes. How am I supposed to know if it's incomplete dominance or if it's multiple alleles? Well, this time, when you've got multiple alleles, you don't end up with a middle phenotype. So this time, what we're going to do is we're going to look at our Drake game, and specifically Drake tails. So if you've been playing through Mission 6, you know what I'm talking about here. So my alleles for Drake tails is you can have a long tail, and long tails are dominant to kinked tails. And kinked tails are dominant to short tails. So I also have 3 alleles, or multiple alleles. So these are not only my phenotypes, but they're my alleles as well. Those are the versions of the gene. Now, we've seen multiple alleles with HLA markers. So in humans, you have HLAs, where you could have like HLA-A, and you could have 1 through like 6,000 different versions of that gene. So you could have all those different alleles, and that's going to make all the different combinations that you can end up having for just that one gene of HLA-A. All right, the next thing that we're going to look at is one that I really like, called codominance. So in codominance, there are 3 or more phenotypes. But all the alleles are shown in the traits. So let's talk about, this isn't shown in the game, but let's talk about chickens. In chickens, we can have black feathers, or white feathers. And the way that this is going to work is if you have big B, then you have black feathers. If you have big W, then you have white feathers. But if you have one big B allele and one big W allele, then you would have both feathers that are black and feathers that are white. So you get a combo of traits. With codominance, you get that combo of traits, and that is actually super interesting. So let's look also at one more, and then I'm going to show you a human example that does a whole bunch of things. So you could have multiple genes. And when you have multiple genes, you have equal probability of getting all of the different phenotypes. But because you could have codominance or incomplete dominance or multiple alleles, it just gets really, really messy. But multiple genes is actually how most traits in humans work. So in multiple genes, you have more than one gene affects the phenotype. And in the drakes, we saw this with drake color. So there was a gene for if it was shiny or dull, and shiny was dominant. And then there was a gene for if there was color or albino, and color was dominant. And then there was another gene for whether or not it was going to be gray or orange. And gray was dominant. So if you had a shiny colored gray drake, then you had those alleles. But you could have a dull gray dragon, but if you had albino, that actually would overrule the other things. So albinos were always shiny, and they were never gray or orange. They were always white. And that's because there are multiple genes impacting how that works. Okay, now for our human example. So I'm going to go to the next page here. And for my human example, we're going to be looking at blood type. And I'm putting blood type in humans because other organisms, like dogs, they have blood types too, but their blood types are different than human blood types. So, first thing is that we're dealing with something that has multiple alleles. We've got A, B, and O as some of our phenotypes. And A and B are codominant. O is recessive. The other thing we're dealing with is you can have a positive or a negative. So this is multiple genes. You've got one gene that codes for A, B, and O, and then you've got another gene that codes for positive and negative, where positive is dominant and negative is recessive. So two genes, gene 1, which is an I gene, immunoglobulin, and gene 2, which is an RH factor. So those are the two different genes. And you're never going to be tested on this, but I think it's interesting, so we're going to use it as an example for how this works. So what's really happening is when you have these different genes, they put proteins on the surface of your red blood cells. So your red blood cells, they sort of look like little donuts, and they carry oxygen. Well, on the outside of them, they have proteins. And they could have, just like Hla markers, they can have these different proteins. And I'm just going to draw a few on here, and then we'll label them. Okay, so the circle one, we're going to call the A protein. The triangle one, we're going to call the B protein. And then the one that looks like a cross or a T, that is going to be the plus protein. And your body can recognize these. So if you have A type blood, you have these A proteins on your blood, and your body recognizes the A proteins and says, that's cool, you belong. But if you have the B proteins or the positive proteins, your body says, get out of here. So each of these proteins is a signal to your immune system of whether or not that cell belongs or not. So let's look at how these work by doing a couple of crosses. First of all, because we have a codominant and a recessive from the same gene, because we have multiple alleles in that gene, we're going to label them. So I is the gene, and IA is the A version. IB is the B version, and lowercase I is the recessive version. So if you have A blood, you can be IA, or you could be IA lowercase I, because the A is dominant to the I. The B is also dominant to the I. So when we're looking at different types of blood, we'll label all of these out. So these are my alleles for this I gene. So A blood is always made from either someone that is IA, or IA lowercase I, that's A blood. B blood is going to have the B protein, and it can have two versions of that B protein or one version of that B protein. Now, some people have AB blood. That's this person up here. And that means that they have the A protein and the B protein are being made in their red blood cells. And then O type blood, that's going to be when you have no protein, so you have the I. So we can make it even more complicated. For our other gene, our alleles are positive and negative. Those are our alleles. So if you have A positive blood, then for one of your genes you have this, and you can be positive positive, or you can be positive negative. If you have negative A blood, then for one of your genes you can have this one and this one, but you're negative because negative means that you don't make that protein at all. So to be recessive, to be negative, you have to not make the protein at all. We're just going to do one cross so that we can see how this works, and we're going to look just at what blood type you have, not this other thing. So I'm going to set up my Punnett Square, and I'm going to make this one kind of wide because writing out all these letters can take up a lot of space. So let's say I have a parent who has A blood, but they have that recessive allele. And then I have a parent that has B blood, but they also have that recessive allele. The possibility of their babies is they could have a baby with A B blood, or a baby with B blood, or a baby with A blood, or a baby with O blood. So they have lots of different possibilities for what kind of blood their baby would be. Now, if we also knew if they were positive, 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 negative, we could also do a cross for that and figure that one out. But I'm not going to, because that's a lot already. Okay, the last thing that we're going to cover is we're going to cover sex-linked traits. Are genes that are on either the X or the Y chromosome. And these can be dominant or recessive, but normally we're going to be talking about genes that are on the X chromosome. So if you have a recessive X-linked trait, then you're going to end up with more boys having the trait than girls. And this is how most of these sex-linked traits are going to work. So let's look at an example from our Drake game where we've got two possible colors. You've got faded, which is the recessive trait, and then you've got deep, which is like a really full, bright color, which is the dominant trait. So because these are on the X chromosome, we're going to write these with an X chromosome, and then we'll do little f for faded. So dominant people could have X, big F. All right, so let's do a cross where we talk about how this works. If we have a mom that is faded in color, so if we have a mom that is faded in color, then we know that she has two copies of the X chromosome. So she has X, X. And then because she has the faded color, we know both of those have to have the recessive allele. She's a carrier if she has one dominant version and one recessive version. That's what we call a carrier. So if we have a mom with the faded characteristic, then what would a dad look like? So a dad that is faded would be X, Y for his chromosomes, but he has the faded gene on that X chromosome. So what we can see here is that in order to have the trait, the faded trait, the recessive trait, dad only needs one copy of it, but mom needs to have two copies of it. And that's going to make it so that this trait ends up in more boys than in girls, because girls can have the other chromosome carrying the dominant trait and therefore kind of masking the effect of that recessive trait. So let's do two Punnett squares to see how this works. So first of all, we're going to have a dad who is deep in color. So he is going to be X, capital F, Y. And then we're going to have mom. And mom is also going to be deep in color, but we're going to make her a carrier. So mom needs to be X, big F, X, little f. So neither of these people, dad or mom, have the trait. They are both deep in color. But let's see how this works. So filling out my Punnett square, always putting capital letters first if they're there. That's a lowercase f. So what we can see here is that based on these are my girls because they've got 2 X's. These are my boys because they've got an X and a Y. So let's look at how many of these are faded and deep. Because both possibilities for the girls have that capital F that they got from dad, that means that 100% of them are going to be deep in color and 0% are going to be faded. On the other hand, boys, because half of them get the big F from their mom, but the other half get the little f from their mom, we're going to have for our boys 50% have that faded characteristic. All right. Let's see what would happen if dad doesn't have the dominant color. If dad has the recessive, so dad is going to be faded. So this boy right here goes on to have babies. He's a boy, so he's XY. Now let's say that he ends up with someone who is not a carrier, so they are a dominant deep for both of these alleles. In this case, we're going to fill out our Punnett square. So this time, dad is faded, whereas before, dad had deep, mom had deep. Now dad is faded, mom is deep in color. And let's see how this works. So X, big F, X, little f. In this case, none of our girls and none of our boys are going to have the trait. So dad cannot give this trait to any of his kids. And this is why, when we're talking about a recessive trait, it's going to show up more often in boys than in girls. Because if girls have it, or if girls are carriers, then they're going to give it to their sons. But if boys have it, they are not going to give it to their offspring unless mom also has the trait. So 0% of kids are faded. All right, so those are sex-linked traits. So when you have a sex-linked trait, you're looking for some difference between the percentages of boys and girls that end up with that trait. So last thing I want to do is I want to go back through and I want to look at how we're going to tell which of these things we're dealing with. So if I have incomplete dominance, I am going to be looking for whether or not there is a middle phenotype. If there's a middle phenotype, then what that means is if I cross a full with a none, so let's say that I have a black bird and a white bird, then the middle type is gray, and all of the babies will be gray. So here's a Punnett square for that, but I used R for my allele. So black, white, 100% gray, or red, white, 100% pink. This happens a lot with color. If I'm looking at multiple alleles, when I have multiple alleles, I have one gene resulting in multiple phenotypes. So this would be like if I had hair type And I could have curly hair, wavy hair, straight hair, or any combination in between. Another example of this is my HLAs. So if I have like HLA-DR, I could have HLA-DR1 or HLA-DR4 or HLA-DR28. There are multiple different phenotypes. I'm going to compare multiple alleles to multiple genes because you might have to figure out if you have multiple alleles or multiple genes. And with both of these, you're going to end up with 3 or more phenotypes. You're going to end up with like a lot of different phenotypes. The difference is that with multiple alleles, I've got one gene and multiple phenotypes. In multiple genes, I have 2 or more genes resulting in multiple phenotypes. So here's an example of that. If I'm looking at 2 genes, hair type and hair color, I get a combo of phenotypes. So I could have a baby that has curly hair and brown hair, curly hair and blonde hair, straight hair and brown hair, or straight hair and blonde hair. But I'm looking for the combinations instead of having one phenotype, but each baby could have a different phenotype. All right, last but not least, we've got codominance. And in codominance, we've got 3 or more phenotypes, but you can have both phenotypes at the same time. So if you cross one phenotype with the other phenotype, you don't get something in the middle, you get both at the same time. So an example of that is like blood type, where you could have A blood and B blood, and if you combine those, so you have the A allele and the B allele, you end up with A and B proteins on the blood. All right, so that is our traits. Those are our non-Mendelian genetics.